Hi, everybody, and welcome back to Let's Combinate Drugs and Devices. I'm your host, Subi Sadeh, and today we are graced and honored by Archana Sah, who is a clinical leader in pharma with over 30 years of experience. She's worked in pharma, biotech, and at CROs, and has driven patient-centered, diverse, and efficient clinical trials. Welcome, Archana.
Glad to be here.
Yeah.
Thank you.
One of the things that has always been perplexing to me, and I just don't understand, is what are the factors that go into designing clinical trials? There's elements around stats and there's elements around recruiting patients. I know recruiting patients has a certain term. It's not coming to my mind right now. There's ethics concerns as well.
Mhm.
And so maybe just broad strokes, talk about like some of the things that go into clinical trials and the factors that you deal with on a day to day.
Yeah, no, happy to start off with that. When we are designing, so when a molecule comes into the clinic for the first time after it has been discovered and gone through the preclinical phase, it enters the clinic. So that's a pivotal milestone point in the life journey of that asset or that molecule. And we start with phase one, which is first in human trials, then phase two, and then phase three, which are the pivotal trials, which based on which we actually submit the data to the regulatory bodies, the FDA, EMA, PMDA, all these global bodies, based on which the drug asset gets the marketing approval. So that's the life journey. We, in early trials, phase one and phase two, the focus is really to understand and first in human trials, phase one, really we are focusing on the safety profile. How safe is this drug? That's the focus of the testing. So there's a lot of PK/PD measurements. There's a lot of biomarker understanding. A lot of it's very burdensome because there's a lot of tests going on from a safety perspective on the patient who are participating. Then we go to phase two, where, in phase one, we might see some early clinical signs where is this effective? Is this drug effective in what we are trying to treat the patient for? So that becomes then the big focus. Once we have understood the safety profile in phase one, phase two, we are focusing on the effectiveness, also learning more about the safety of the product, but in larger number of patients than what we did in phase one. And then once the drug clears even that in phase two, then they enter the phase three, which is a pivotal registrational trials. And that's where the focus then is a lot about, is this effective in a diverse patient population? Is this effective in the target patient population? And that's when, in the design of the trials, the comparator arm becomes very important. Because when in your protocol design, I've spent 20 plus years out of my 30 plus years in clinical development in the oncology space. And for us, we always have to compare and show that the new drug that we are trying to bring to the market is better than the existing or the standard of care, as they are called. In the right phrase, there is better than the existing options for that patient. So when we typically wouldn't do placebo trials, so instead we would test our drug against the standard of care. For example, let's say if it was an immunotherapy drug that we were researching and bringing into the clinic through approval, we would compare that with, let's say, chemotherapy, if chemotherapy was the standard of care for that kind of cancer. Or we would compare that to a targeted therapy, if that was a standard of care. Sometimes it's a combination. Now, in immuno-oncology especially, where I've also spent a decade of my time, I can say that in immuno-oncology now, we used to start with combination therapies and then third line, second line therapies. That's the other thing that your audience should try to understand is, in oncology, the other complexity that comes into play is patients that have usually failed their first, second, and third line of therapies. That's when they enter clinical trial because there's no other option left, no other molecule that is already marketed out there that they can, because everything else has failed. So often they are in their third line therapies or second line therapies when they enter a clinical trial. So that's the other thing we have to keep in mind when we are designing these oncology trials, clinical trials, is the line of therapy as well as the comparator arm. So the comparator arm will be very dependent on what is the standard of care. If, let's say, for an and I'm just going to say lung cancer, for example, let's say if there's an immunotherapy plus that we are comparing in combination with chemotherapy against, there could be three things that could be the standard of care in terms of chemotherapy, or just one thing like a platinum therapy or a chemotherapy as an example, that is a standard of care. So these design aspects are very critical And we have to discuss that with the FDA, or regulatory bodies in whichever region we are working, going to be marketing this drug. And that design has to be acceptable to the FDA before we can ever dose the very first patient. We get that clearance from the FDA. By the way, even the phase 1 and phase 2, they also have to go through that. But we have, when first in human trials, those are also go through the clearance before we can dose the first patient. That's just very broad strokes in terms of how. And then the clinical trial, actually, just from a protocol design perspective, we also have to be very careful of what's the disease area, what's the prevalence of the disease, where are these patients located across the world. So accordingly, we'll actually select the site and select the countries that will be participating in the trial. The next layer is, what is the heart and soul of the protocol is actually the schedule of events, or the table where we actually call out every single patient visit and what tests will be done at each of those visits, because that gives us the most critical data. Data is currency for us. So the data that we are going to be submitting to the regulatory bodies, that's captured in that schedule of events and is the heart and soul, so to speak, of any clinical trial. That's where we define what we are testing, how is that being tested, what data is being collected, what will be the primary endpoints into the protocol, which will be what the regulatory bodies will have had prior agreement with, and what will be the secondary or exploratory endpoints, because we want to also learn about the more mechanism of action of the drug. Thirdly, we also will have a patient reported outcomes, or PROs, as they are known in short form, which are very critical from the regulatory perspective, because the FDA and other bodies have said that just the overall survival or the efficacy of the drug is not important. Equally important is how did the patients feel? How was their adverse events profile or their safety profile while they were going through this treatment on your protocol? So that is, they want to measure that from a clinical perspective, so using lab tests or imaging, etc. But they also want to image the patient's response from their effect on daily life. So those are measured through the patient-reported outcomes. That are also, there are standardized forms for those, which are used for collecting the patient input as well. And then really, once the data is collected, the enrollment is a big part. There's a startup phase, then there is the enrollment phase, the selection of, and then there is a closeout phase, and then there's a database lock, and the data gets submitted. You mentioned about the vendors, I think. So I think there's a lot of clinical vendors that are selected that will carry out different key aspects of the protocol as a study is being run. For example, there could be a lab vendor that's going to do all the lab tests. There could be an imaging vendor that's going to do all the imaging and collection of the imaging data. Then there could be digital health components to it, like a consent provider or a ePros, a patient-reported outcomes provider. There could be other ancillary elements going on in the protocol, like remote health nurse. If you're going to be doing a decentralized clinical trial where certain tests and procedures are being done in the patient's home or closer to the patient homes in a clinic or at a pharmacy, you have vendors that will provide those facilities and others that will be done at the clinic. For example, imaging, you often do that just in the clinic, and there are not portable tests very easily available for that. And also, the patient preference is another thing we keep in mind when we are designing the protocol. Throughout my career, one of the things that I've noticed is there are ethnic and cultural differences in the preferences of the people. For example, the African American community, they don't like welcoming strangers into their homes. Like when we would send remote nurses to take the blood for lab tests, they prefer to go to the clinic instead. But when you look at the Asian population, they preferred not having to go to the clinic, but whatever tests can be done in their homes, that was their preference. So we cater to all of these when we are designing the protocol and the tests and the complexity that comes in from the different vendors that are selected. And of course, the statistical aspect is huge because that will decide what data we are collecting, how the data will be analyzed, so that there is very clear answer of whether the drug or this combination of drugs that are being tested works from a safety profile as well as an efficacy profile.
You touch on a bunch of different factors there. I think one thing that's always been interesting to me whenever I've heard anyone talk about clinical trials is this idea of first line, second line, third line. Where are those actually written? I know that as far as if something is indicated, doctors have flexibility to
Yes.
essentially prescribe whatever they want. Is this on a formulary? Like, how do you know what's the first line treatment versus third line and what your
Yeah. Yeah. That's such a good question. I think so. Depending on when you're bringing a drug into the clinic, there's one, I think you had one episode on the CDP, the development plan of the molecule. And one of the key aspects of that CDP is going to be what is the competitive landscape? What are you going to be need to test against? And what usually in oncology, the most of the molecules when they come in, they need to be proven in terms of in the patients are dosed with them usually in the second or third line. In other words, some standard of care has failed. There are and then this is the next best option for the patient. So then the investigator wants to treat the patient with this next best option if the standard of care failed. That is called the second line therapy. Let's say they tried two things. They tried first immunotherapy, then they tried combination of immunotherapy and targeted therapy. Both those failed. Both of those didn't work on the patient. Then now they're then their next is best thing for them is a trial. That then becomes a third line therapy that they are trial. And this is written out in your clinical development plan, in your protocol. It's very clear to the investigators when we give the protocol to them what is the target patient profile that meets the inclusion exclusion criteria for this protocol. So it's very clearly defined in the protocol
but
which patients are going to be recruiting.
and so I know that part, and actually for if you care, like with combination products, the patient population that the molecule is targeting ends up being the basis for a device combination product's user needs, and that's what we use to define. For example, if for example a product is used for arthritis, let's say,
Mhm.
that would be the input to how we design. So a pen design would need to accommodate the requirements of somebody who has
Yes.
arthritis or small hands or whatever. I guess what I'm picturing is a funnel where, if somebody is in a disease state and they have options of care available to them, what's currently available as like the common treatment is obviously what you would try first. If that didn't work, then you
Mhm.
try like stuff that is available but is less commonly used, whether it's expensive or less available, whatever the reason is. And then after
Mhm.
that, you would go to something like a clinical trial. I understand that. But if you're on the clinical development side and you're trying to
Mhm.
figure out how do I enroll patients, part of your selection criteria is they need to have tried A and failed, and then they need to have tried B and failed
Yes.
before they get to C.
Yes. That's a
Now,
patient pathway.
okay.
Yes.
Patient pathway is like
That's a
the
patient
funnel.
pathway.
Patient pathway.
Yes, exactly. What you're describing is a patient pathway for that disease.
Yeah. Now,
What is the current patient pathway? Yes.
I think what I want to understand is the standard of care. How do you know what that is? Because doctors
Oh,
essentially
we
can
go
give
through a
whatever they
lot of
want.
yes. No, there are NCCN guidelines. There are
NCN.
all these guidelines that actually define standard of care for oncologists, for example, in oncology. That is so when doctors prescribe, when a patient with, let's say, a newly diagnosed lung cancer shows up, that there are guidelines as to what should be the first line of therapy. Second, so I think there are guidelines that are followed, and that is what the standard of care constitutes the standard of care. So it is now there might be small, minute differences between some regions in the world because of lack of approval of that standardized care. Let's say that is NCCN guideline that says you should give this drug as a first line of therapy, but that drug may not yet be approved. Let's say in Peru or Argentina, then in that case, in those countries, the standard of care may differ. So when the patient is because of lack of availability of the drug, either from a cost perspective or from lack of regulatory approval, and hence it's not marketed that in the country either. A lot of these discoveries happen. The pace of discovery and pace of innovation in terms of better options available for patients sometimes may be faster than the regulatory approval available in those countries. And oftentimes it's the social determinants of health that impact the lack of availability of that drug in those countries as well. By the way, when we are designing the trial, we have to understand which countries we are going to go, and we have to understand the standard of care in each of those countries. And oftentimes, we actually ship the standard of care and provide it to the clinical trial site who's running the trial so that it's available to them, which they may not be able to afford given the high expense or the lack of regulatory approval. Yeah.
You talked about some elements of clinical trials that are related to ethics. I know your background is oncology, which has its own ethical kind of
Mhm.
situation and concern. I've worked in vaccines before where I remember just I'm not really involved in the clinical piece and definitely wasn't at the time, but we would know when the readouts are and we'd know about some elements. And I remember hearing them say, we need to make sure we have enough clinical trial material to be able to dose appropriately our drug, but
Yeah,
we also need
yeah.
to find our competitor's drug so that we can dose it as well, because it would be unethical to have a
Yes,
control group for something that
yes.
people may be exposed to and not give them something that we know works as a standard of care. And so my understanding, and this could be wrong because again, I'm not a clinical person, is that in the vaccine space, you're comparing how much better are you at protection versus what's currently available. That's my understanding. Is that correct?
And also, even in the oncology space, it's the same. You're compared to what's currently best available treatment or standard of care treatment, as I defined earlier, for the patients. So that same logic applies even in the oncology space. There are certain placebo-controlled trials in other therapeutic areas, other than like when I ran dermatology trials, for example, or some I did have a placebo medication that would look, we would make sure that it looked exactly like the tube that had a cream, for example, which the active ingredient for psoriasis looked exactly like the tube that did not have the active ingredient, but had all the inactive ingredients. And hence A versus B was compared in that psoriasis trial, for example. But in oncology, we usually compare to the standard of care. And I think you have to remember at the end of the day, these are oncology patients. They're fighting for survival of their lives. And the standard of care cannot be no treatment. So the standard of care has to be, the comparison has to be to the standard of care, which is the best available treatment currently as per the guidelines and as per the prescribing norms that patient has availability to.
And so I think what's painting a picture is that certain physicians, depending on the area that they work in, they have their own consortiums and they have their own papers that
Yes,
they
there are
use
these
to.
group bodies. There
And
are national bodies,
yeah,
actually.
yeah.
Yeah,
These are
actually,
these
they're deciding.
conferences that are like 70,000
Yes.
physicians or whatever. They're huge.
Yeah, the
ASCO,
ASCO that happens
exactly.
every year, the ASCO guideline, it happens every year in
It
Chicago.
was in Chicago,
There are
yeah.
ASCO guidelines. There are, so all of these industry organizations, they put out ASCO recommendations, ASCO guidelines, or NCCN guidelines. The key difference is also from a payment, right? Reimbursement, especially given the health system in America, the reimbursement of certain drugs happens if they are on the NCCN guidelines list as well.
Really?
And hence, yes, then hence, so you'll see there is also the financial aspect that comes into play sometimes, which is in some, but for the larger part, I think the key when we're designing the trial is we are going head to head against what is the standard of care or the best possible option currently marketed that is available for this patient. That's the mantra that is used, and that's what is acceptable to the FDA. Except FDA will not accept subpar comparisons
Mhm.
and will not give you approval to dose a trial, to dose a patients in that trial if you are comparing it to a subpar comparator arm, and which may give you an easy win from for marketing that drug, but FDA will not approve that because you have to be competitive. You have to be competitive to what's the best option out there for that patient.
And that makes sense because otherwise you have just a bunch of options that like more or less either work the same or I can get behind that. I definitely want
Yeah.
to talk about the future of clinical trials, but there are a couple questions that are still like not super clear for me. So one of the things is like the different types of clinical trials. We always hear about randomized double-blinded studies. BABE study is like the other one that I think is common. Could you maybe walk through the different types of high-level clinical trial designs and like when and where they're used?
Yeah, I think basic is this is when you're doing the designing the clinical trials, you could have double blind. It could be double blind design. It could be the randomized clinical trial is an important word I want to start out with. So that is the most robust type of interventional trial where patients are randomly assigned to treatment or the control group. So that's very clear. Then we have things like design, like in oncology, there are because there are so many different kinds of types, lung cancer, breast cancer, it's based on the location of the body part. There are different kinds of cancers. And then there are the other aspect that we keep in mind is umbrella trials, basket trials, and multi-stage trials. So I want to define these for you. Most common type of clinical trial where we are testing the new treatment, the new combination of treatments. So a basket trial is a type of trial where they group the patients based on the genetic mutation of the tumor. So regardless of the cancer type, they test the same drug on all of them. That's called a basket trial. An umbrella trial will be a trial where one type of cancer that the group of patients that groups patients into different sub trials based on the genetic mutations in their tumors, each testing a different drug. So remember, in the first one, I said the based on the genetic mutations of the tumor, regardless of the cancer types. And the second time, the second umbrella trial, where we're grouping the patient into different sub trials based on their genetic mutations, each testing a different drug. Basket is regardless of cancer type. Umbrella is regardless of drug. The multi-arm, multi-stage trials, those become a little more complex. Those then trials have a single control group, but multiple different experimental treatment groups, allowing researchers to efficiently test several new treatments simultaneously. These bring a lot of complexity, by the way, into the execution of the trial, but they are needed to understand at the early stages. Especially, you'll find these designs a lot in the phase 1 and phase 2 area time frame, where they're trying to understand multiple different aspects of the mode of action of the drug, the safety profile of the drug, the efficacy of the drug in different combinations. So those come into play in a lot of our oncology designs. And of course, I already talked about the phase 1, phase 2, phase 3. And on phase 4, I just want to clarify for your audience, those are the post-marketing surveillance trials, where once a drug is approved, it is available to the public. Those trials continue to track the safety, effectiveness, and the optimal use in the real world population, in the general population. Those are the phase 4 trials.
That last one that you were talking about, I know is commonly looked at as like a real world evidence trial.
Yeah.
Um,
And then there are multiple blindness also. They could be single blinded, they could be double blinded, they could be triple blinded. So that's another dimension of the design as well, or a quadruple blinded, which is very rare. But I just want to, from a blindness perspective, that's the variety you have there as well, depending on the need of the protocol. Yeah. So that was the other
yeah,
aspect.
and,
Drop in.
and that also has impact on how the clinical trial material is manufactured, and like
Exactly.
and that's obviously a consideration when doing audits of and qualifications of sites that are doing that kind of material.
Yes.
Okay, you can make material that's to specification, but can you handle clinical labeling, clinical packing, and stuff like that? And that's where there are some
Yes.
companies that are well
Yes.
known where their like specialty is clinical packaging. Now, are BA and BE studies the most simple studies?
The bioavailability and the bioequivalence studies, they are, well, I do want to emphasize how important they are, first of all.
Yeah.
But they are often done in phase 1, where we are looking at the PK/PD profile. They assess how the drug is absorbed into the bloodstream, how quickly it reaches the site of action, how quickly it exits the body. So those studies are really important as a fundamental and a part of every regulatory package. They're a very key requirement for the regulators to ensure that the comparability between a pharmaceutically active test compared to a reference item that they are looking at the data for. So they are absolutely important. They are defined and conducted in the earlier times, and they form a very important part of the regulatory package, which the rate of absorption and the rate of excretion, the at which the active molecules from a drug product is a very vital information, because that is directly linked to the clinical efficacy as well as the safety profile of the drug. So those studies are crucial. Without it, yeah,
Yeah, I know they're obviously super important, and we have to do with them a lot of times when there's a concern about bridging of this went through
yes,
we had a vial that went through clinical trial and we're putting in a pre-filled syringe or it's
yeah,
whatever. We want to make sure that the fact that we put it in this delivery system doesn't impact its ability, and so we don't have to do a full-on clinical trial, but we have to do a BABE study. Okay,
okay, basic.
but I think what I'm more asking is like in terms of clinical coordination, are BABE studies the least complicated?
They are relatively much less complicated, but they have a lot of There are a lot of stringent measurements that they are being done. Like, for example, when you're doing a PK study, you would need to have a patient in the clinic for a period of 8 hours where you're withdrawing blood every half hour or every hour, depending on the pharmacokinetic profile of the drug. So it's very labor intensive for the clinical staff, as well as very burdensome for the patient as well. So what we have for that is we actually have dedicated phase 1 clinical units, clinical testing units, where they have all the infrastructure to bring in the patient, keep the patient comfortable for that long periods of time, the infrastructure to be collecting all these different tests like blood pressures, blood draws, all vital signs, all of those are measured very far more frequently during those studies. But the sites are very equipped for that. And you also have to have an ER, management of emergency. Let's say if the patient experiences an emergency, what infrastructure you need within the hospital itself to manage the emergency situation. So these are very specialized, dedicated units. In addition, sites that do regular phase 2, phase 3s also have phase 1 units because they have those specialized capabilities and it is inbuilt within their site. So they can do your phase 1s and beyond as well.
Yeah, and that's where things like SERs and incident reporting and all that stuff comes in, where, like, for
Yes.
clinical, it's obviously super important. Now, transitioning into kind of the future of clinical trials, um, I'll start off by asking, in the current climate that we're in, where there's like a mistrust of institutions and stuff like that, there's this perception that clinical trials are designed in a way that's manipulative. Could you maybe talk about that and what's needed to maybe deal with that perception? And then moving in, I
Yeah.
want to talk about like the future of decentralized trials and remote patient monitoring and AI and all that like new
Yeah.
stuff. But I think I just wanted to start by
Yeah.
saying our current state, to do all that stuff, we need to get over this like cultural hump of, oh yeah, they're just
Yeah.
designing the trials to say whatever they
Yeah.
want.
Well, that's where all these safety check-ins come into play, right? When we design a trial, we have to get in front of the FDA and justify the design and in order to get that design approved before we can ever dose the very first patient. So the integrity of the data, we are defining what data we are collecting, how it's going to be collected. All of that is predefined. So the chances of manipulating data or manipulating the design of the data. And I would question, first of all, the motive, because oftentimes the science has to come through, right? The experiment that you're doing has to follow a scientific path. There's a statistical analysis plan already predefined in that protocol. So the opportunity to fudge that or change that is not there. It's all very clearly defined before you got in front of the FDA and shared your protocol with them for approval. You have predefined all of that. Now, one area where, so before I comment on that, I don't believe that there is opportunity to fudge or commit fraud if you have put together a very solid design and that has gotten in front of the FDA. There are these checks and balances in terms of what they're evaluating before they're approving it for you to dose. You have to also remember that every trial all the pivotal trials before the approval, when after the data is submitted to the FDA, inspectors come and inspect. That's part of the FDA inspection. And in that portion of the clinical trial life cycle, the FDA is looking at what data was predefined with the FDA. How did you conduct the trial? Was it done with quality in terms of the patient monitoring data, in terms of what you said, how you would collect what data? Was it followed? In other words, they will go through all the detailed inspections. So they actually inspect the study file. They even go to the sites that participated in the trial and collection of the data. They inspect the sites. They interview the doctors who conducted the studies as well as the staff. Then they'll come to the sponsor site and they'll interview the sponsor team that actually conducted the study and look at the data. So there are all these checks and balances where there's very little opportunity to to commit fraud or do a subpar study or change anything from just now. Remember, if it is, let's typically in any protocol goes through on an average 5 to 6, sometimes 8 to 10 in oncology amendments, like changes. So even if those are significant changes in amendments, we have to go back in front of the FDA and submit that amendment and the IRBs as well, but
Yeah,
from a
that
patient
takes work.
protection perspective, to make sure that those checks and balances are inbuilt. To be honest, so yeah, I have full faith Now, the one area where I would question a lot is, let's say if there were not a lot of diverse patients enrolled when you were testing. Now, historical data has shown that 80% of data collected for all the medicines approved where the trials were done on white male population specifically.
Really?
So then diversity, yes, but then the diversity of the trial is not there. And then that is why when they are then prescribed in the real world, oftentimes they're ineffective in certain patient populations because diversity of the data was not enrolled during the testing phase of that drug. So that's the area which I would question more. And that is one of the key reasons FDA has now, and the Congress in the United States has actually enacted the DEPICT Act in December of 2022, where now for all pivotal phase 3 trials, every company has to submit a diversity action plan, also known as DAP, where they actually call out what diverse patients they're going to be enrolling to show the effectiveness and the safety of the drug in diverse patient populations. Plus, they're going to have to call out how are they going to enroll, what tactics are they going to use to enroll that diverse patient population. And believe it or not, the FDA is now required by Congress at the end of every year to submit a report, to publish a report where they actually see whether after this act, the DEPICT Act, whether the companies are actually doing that or no, and what is checks and balances. So the FDA has to put out a report every year on that. So under the current political environment, it's a little bit difficult to, but I can certainly say that because of the DEPICT Act, sponsors are still required to submit that diversity action plan. And when you think we talked about ethics earlier, right? From an ethical perspective, it also makes sense that you test the drug on a diverse patient population
That's representative.
because eventually you're going to be marketing them to the diverse patient population. You want to make sure that it's safe and effective on and diversity is multi forms of diversity, just so you know. We're talking about not just the age diversity, the gender diversity, we are also talking about socioeconomic and social determinants of health diversity. It no longer should a zip code determine the zip code of a patient determine whether their access to innovation and the best medicines out there. We are also talking about the socioeconomic diversity as well, where we are enrolling patients from the remote parts of the country where the socioeconomic and access to innovation may be very different than, like, say, people living in Chicago or New York or San Francisco, right? So we want to make sure we are enrolling patients from diverse patient population, and that's where the community comes. Testing a drug in the community setting, in the rural setting, comes into play. And that's a perfect segue for me to get into the next question you asked, which is how are these decentralized clinical trials that we talk about, the hot topic for the past few years, these are vital technologies that are enabling us to take the trial to where the patient resides in the remote parts of the world, in the remote parts of America where the patients reside, as opposed to every time having to bring the patient to the clinic. Even for a vital sign measurements, the patient had to undertake the arduous journey of 2+ hours or 4+ hours, the total of their time. This was very burdensome, both physically as well as financially, as well as from their other jobs that they couldn't take time off. So, in an effort to boost this diverse patient enrollment, the decentralized trials play a key role where now we can only bring them in for, let's say, 50% of the procedures. AstraZeneca, I presented this in my presentation at the conference where we met, where AstraZeneca had looked at 91 different protocols and ranging from oncology, respiratory diseases, cardiovascular diseases, multiple different therapeutic areas. And they came to a conclusion, you'll be shocked, that 40, I'm going to look at my exact statistics here. So 91 clinical protocols in oncology, respiratory, and cardiovascular disease, where that 74 to 85% of trial assessments could be successfully collected remotely using clinically validated devices.
Mhm.
And that would result in a 40% reduction in the number of physical visits to the clinic. Imagine the impact of that. That's the transformative potential of decentralized clinical trials and digital health.
So with
Um,
because I started hearing a lot about decentralized clinical trials right around and a little before the kind of COVID pandemic is when I think maybe in the late teens, 2017, 2018, people started talking about it when everything was shut down. People really started talking
yes,
about it, and it's been going on ever since. I
it was
guess
a lifesaver.
I guess where like I'm maybe trying to draw a parallel with where companies are at, where everybody went remote and then hybrid, and now there's a lot more return to site. Is there that kind of similar situation where decentralized were thought of as something that was really important and critical, and now people are like, maybe patients don't mind going to the clinic, or maybe the readings end up being more accurate, and there's a saying in Arabic, like, I don't know how I don't know how to translate it perfectly, but it's you it doesn't have to be either this or that. Maybe it's like a little bit of this or and a little bit of that.
Yeah, yeah, yeah. Yeah, yeah. So you bring up a good point. So during the pandemic, as you mentioned, it was a lifesaver. Suddenly overnight, that was the only way we could maintain the continuum of our trials, because we had to deploy these decentralized trials to take the trial to where the patient's home was. A lot of the critical phase 3 pivotal studies, they almost overnight we had to get these deployed on those studies, and in order to maintain the continuity of collection of very critical endpoint data. I want to call out, and to answer your broader question, I want to call out this is real. So, Tufts University, they're backed consortium. And there's another thing I actually presented at my presentation at the conference when we met. They presented data where they looked at 69 total trials with database log from January 2018 or later, for which the data was collected from 14 pharma companies. So each pharma company, on an average, provided data from 5 trials, and data was compared between trials with DCT versus no DCT.
Mhm.
Okay. And they examined how much of the total trial procedures and visits can be conducted remotely, what DCT solutions were being utilized, and they examined the correlation between DCT solutions and participant diversity. Remember, that was one of the added bonus of DCT, right? We were able to suddenly now enroll diverse patient population. And there were phase 1, phase 2, phase 3, phase 4. All of these trials were part of the group, and they were anti-infective, cardiovascular, CNS, endocrine, GI, immunology, oncology, respiratory, all these therapeutic area trials. So good mix there. And they found that the study visit activities, 88% of trials from that group used one or more DCT solution for each activity. That's pretty high. Now let's go one step deeper, right? Let's look at what was this DCT being used for? They were being used for wearables and sensors to do remote patient monitoring. They were being used for different apps for data collection, for example, patient reported outcomes that I talked about in the earlier part of this podcast. They were being used for apps for reminders to patients to complete their patient reported outcomes, or reminders about their upcoming visit, or reminders about taking the drug in their home if the drug was a pill and was shipped into their home. They were being used for home visits. When we sent the nurses to the patient's home, they were being used for that. They were used for mobile or pop-up locations, oftentimes close to a large metropolitan area, close to a rural area, community center. Some pharmas even sent out mobiles with pop-up locations. Vans inbuilt with a lot of these capabilities to measure things the same as what they would if they were going to a clinic site. And they found that trials with DCT solutions, and this is something I touched on earlier, where Asian population had a higher percentage of participating patients. Black participants had a lower percentage of participating. And luckily, they found there were no differences were detected when devices were used. In other words, all ethnic populations treated devices the same way when they did remote patient monitoring. And it was very critical to show that a lot of these different decentralized trial techniques that were being used, and by the way, FDA and a lot of other regulatory bodies, they are asking and they are wanting to see all of these decentralized methods being used, because that's the only way we can take all these innovations to the community setting, enroll diverse patients, understand the safety and efficacy profile in multiple different ethnic populations. And these decentralized trials are being used. Now, the flip side of that, I do want to talk about that too. The flip side of all of these wonderful things being used is the complexity it brings in the execution of the trial, the complexity for the sponsor or the CRO who's running the trial, the complexity of multiple vendors you need now who are providing different solutions for all measurement of all these things remotely, plus the complexity of the data collection and management of the data, analysis of the data, right? So they have to follow all these. So the complexity from a conduct increases for the sponsor or the CRO, but bringing in these hybrid approaches where when I coach CEOs and teams that I work with, I tell them you have to deploy these DCT solutions in a very fit for purpose approach. And You cannot do a one solution fits all. Like you said, right? You, if, let's say, in what worked in America may not work in Argentina or Peru or Southeast Asia, in other words. So you have to deploy these in a fit for purpose approach and get a right balance of complexity management versus the key important endpoint data collection without increasing the burden on the sites and without increasing the burden on the patients. That's what I define as fit for purpose approach.
I have seen like we have to do a lot of human factors testing when we do our device development, and I have seen situations come up for those where we have a lot of trouble in recruitment, and we'll go to FDA
Yes,
and request, could we have flexibility about the inclusion exclusion criteria where, you know, maybe they don't meet all of these specific requirements, and here's the rationale and justification, because we're only trying to evaluate how are they using the item, not clinical efficacy, more like their interaction with the product, following instructions and stuff. And maybe we can broaden it out to include the
yes,
patients and caregivers for these three disease
yes.
states instead of just this specific one. And
Yep.
we have been in situations where they just said no, no, you can't. Now, when it comes to
Yeah.
human factors, it's hard to do I think it would be hard to do decentralized because you need to control environmental factors and you need to make sure that those things are. However, if you're in that situation with a clinical trial, you can find people wherever they are. And ultimately, I think your target patient, that's, let's say you have a very specific type of cancer, you're not going to find tens of thousands of people that have gone through the
No.
patient pathway that are
Yeah.
available. It can be on your trial. And so you'll take them wherever you can. Right. And so I think
Yes.
that's where
Yes.
some of the tools that you mentioned become really helpful, but
These tools come in really helpful then when in order to going to where these patients are.
that doesn't mean that like somebody like me who lives in Chicago and lives like less than 10 minutes away from Loyola Medical Center, maybe I don't need that. Maybe I could, like, if I was
Well,
ever to,
that's the flexibility. And
yeah.
that is so, in addition to fit for purpose, the other advice I give is that you have to give the flexibility. You have to build it into the protocol where if a patient only wants to go to the clinic, they have that option. If the patient wants to do 3 out of 5 procedures into their home and only go in for 2 or the other way around, they have that flexibility. So the flexibility is another thing that is built in, needs to be built in within the protocol. And within the protocol and also in the data analysis plan, because what data will come from where and feed into the clinical database and how's that so that has to be in parallel, absolutely.
All right. The last thing that I wanted to talk about is technology and regulation. I've worked on some things that were pre-IND, and there's a lot of
Mhm.
things that go into an IND application. I think if anyone did basic research about what all goes into getting the go ahead to put clinical trial material into first in human, I think most people would be like genuinely shell shocked at how much work goes into getting that approval, the back and forth, all the different stakeholders that need to be involved, internal and external, outside of
Yeah.
regulatory bodies. Are there
Yeah.
areas that you feel like in industry that need more regulation, more tech? A lot of times technology is a product trying to find a problem to solve, but are there real problems that need more technology or areas that need regulation in clinical trials that are currently not as well regulated as they should be?
Speaking from the clinical trials, like once the molecule enters the clinic, not the preclinical side, but speaking from that side, I think there is always a place and for technology and all. Now of course AI is being used in a lot of those areas where we can bring impact a few things. One is efficiency into the process. Number two, timelines. We can impact timelines by making the collection of the data faster. We can. So, from my perspective, I think when I think about the entire clinical trial life cycle, the extent of adoption of the technology into the clinical trials varies from sponsor to sponsor, varies from the capability, understanding, willingness, because it's also expensive, right? It brings complexity, but expense along with it. So all of these factors feed into the adoption or lack of adoption, so to speak, into the clinical trials. From a regulatory perspective, what I can tell you is that regulators do want to see the deployment of these technologies in a very fit for purpose approach. Depending on the inspector or the regulator you're working with, you mentioned, you gave an early example, oh, we wanted to do this in the device area, but we were told no. In those instances, my advice to the teams would be engage in a dialogue. Just don't take no for an answer. Engage in a dialogue with your regulator as to why they think that they cannot allow you. I've been at several conferences, even at Scope this year. I can tell you that there were different discussions in the rooms where the regulators were in the room and they said, no, I mean, what we are looking for is not a blanket. You can only do one thing and you can do only one thing across the entire trial. As long as you come to me and you show me I'm doing this for this population because this is what makes sense in a fit for purpose approach, and then I'm doing this for this population, and there is integrity of the data being collected, we do not say a blanket no to deploying a lot of these technologies. You have to maintain the integrity of the data. You have to justify why you're deploying this. So engage in a dialogue. Don't just take no, and if they said no, don't just go away. Engage in a dialogue with them. Draw out the FDA guidelines. There are very specific FDA guidelines on deployment of a lot of these technology and tools. The DCT guidelines, for example, they get into it in depth. And there's very distinct FDA guidelines now that have come out that shows us that the FDA wants us to deploy a lot. Real world technology is another thing which we haven't talked about. How can real world data be leveraged in clinical trial development? But that's another area where how that data is being analyzed, how that data is being deployed in the protocol design, all the way through analysis and follow-up. Tokenization is another thing where using tokenization of the patient data, we are able in oncology to follow that patient even in follow-up after they've finished the clinical trial. What in future, what are the other, let's say the disease is gone and then it comes back after 5 years, what therapy did that patient get onto at 5 years? In cell therapy, by the way, we follow the patient for 15 years. There's a lot of technology that is being utilized, and the FDA and the regulatory bodies are very much for it. If they are ever against it, my advice to your audience would be get into a dialogue with your regulator that you are having the discussion with. Ask for a meeting, ask to meet them, and show up with your having done your homework with a lot of these FDA guidelines that justify the use of what you're doing.
Archana, where can people find you?
I am available on LinkedIn. My email contact there, posted on my LinkedIn profile. That's the best way to find me.
Awesome. Thank you for coming on, Archana. This was great.